Hi friends, welcome to classes by Lab Doctor. This is Dr. Divya, and in today's class let us continue with the lipid transport. So till now we have seen that this lipids, they being a very important source of energy, how the dietary fats are digested in the stomach, then how they move into the intestine, how the chylomicrons are formed, how the chylomicrons are absorbed into the bloodstream, and how they are utilized by the peripheral tissues, and how the chylomicron remnants are there, and how they are also taken up by the liver, and it is metabolized there. Moving ahead, we will see that the liver is a very important organ in the lipid metabolism, and this liver is the primary organ responsible for synthesis and secreting the VLDL. VLDL is another lipoprotein which carries your triglyceride, and these triglycerides are carried to the peripheral tissue where they can be utilized. So this triglycerides will be providing this fatty acid to the peripheral tissue, and these fatty acids, they are utilized for formation of the membranes, for formation of hormones, various other functions. So this is the VLDL molecule. Later on it will be broken down into IDL, that is intermediate density lipoprotein, and then the LDL, that is low density lipoprotein. So now the VLDL is getting converted to LDL, and in between you have the intermediate density lipoprotein also. So VLDL is synthesized in the liver, and what is the main function of the VLDL? It is a lipoprotein, it is a van, it is a carrier which will carry the triglyceride to the peripheral tissues. As VLDL will travel through the bloodstream, the muscle and adipose tissue, they will extract this triglyceride, causing the VLDL to shrink, and then it will become the intermediate density lipoprotein. So VLDL will become IDL. So what happens is, this car carrying this triglyceride, when it is traveling, the VLDL car, when it is traveling in the bloodstream, it will deliver it to the muscles, it will deliver it to the adipose tissue, wherein once the triglyceride is taken away, this VLDL molecules, the density of which will decrease now, and it will become intermediate density lipoprotein. So this intermediate density lipoprotein can be further metabolized to low density lipoprotein. So this LDL is the end product of VLDL metabolism. So now LDL, the triglyceride has gone, and now it is having more of a cholesterol. So higher level of LDL, you all know it is a bad cholesterol. It is having a increased risk of cardiovascular disease, atherogenic. So VLDL, LDL, these are all termed as a bad cholesterol because they have a potential role to play and affecting your cardiovascular health. So here you can see in the liver, the VLDL is formed, and from VLDL you will get the LDL. So this I have already explained. The chylomicron was dropped from the intestine, and the chylomicron had this ApoB48, which was used to drop this chylomicron from the intestine. This chylomicron was given ApoC and ApoE by the HDL, which is a good cholesterol, and ApoC and ApoE were given so that ApoC will activate, or ApoC will cleave and cut the fat, and also it will help in the activation of this lipoprotein lipase, so that it will break, it will cut and cleave these fats. And ApoE was very important in the endocytosis, in eating up the remnants, chylomicron remnants. And here in the liver you will see that the VLDL is synthesized, and VLDL is having ApoB100 protein. So this VLDL, now what happens is, this VLDL will be converted into LDL, and again here you will get the role of HDL. We will see it in the further slides. VLDL, and what is the role of this HDL in this VLDL? So VLDL, I have told you that liver is the one which will synthesize this VLDL, and VLDL is rich in triglyceride. So as VLDL circulates in the bloodstream, the lipoprotein lipase enzyme, which is seen in the blood vessels, they will hydrolyze the triglycerides in the blood, in the VLDL. So then what happens? From the VLDL you will get VLDL remnant. So from this, the hydrolysis process of this VLDL remnant will give rise to intermediate density lipoprotein, which are lesser density compared to VLDL. And this intermediate density lipoprotein can be taken up by the liver or further metabolized into LDL. So this IDL can be again taken up by the liver, or it can be converted into LDL. And this VLDL remnants and IDL acquire apolipoprotein E from the HDL. So HDL is the one which will again give them the ApoE. ApoE is what? Is that apoprotein which will help in the endocytosis to take up, to eat up all these remnants whatever are there. It will see that it will not leave it in the tissues. It will see that it will carry it back to the liver. So HDL. So this H is standing for hepatic delivery. So it will do the delivery to the liver. It will take this whatever remnants are there. It will help it to go to the liver. So that is why HDL is clearing it up, all the bad things. So that is why it is good. LDL, what it does? LDL will carry these cholesterol to the peripheral tissue. So it is loading the peripheral tissues with the cholesterol. So LDL level, if it is increasing, that is why this is called as a bad cholesterol. So LDL will deliver cholesterol to the cells, and HDL, what it will remove? It will remove all the excess cholesterol. It will remove all the remnants, and that liver converts this one. The liver will handle the remnants. So that is why HDL is the good cholesterol and LDL is a bad cholesterol. Here you see this was VLDL. This is a VLDL remnant. It was having ApoB100. Here also there is ApoB100. But now the HDL has given it ApoE. So what ApoE will do? It will eat up. It will cause endocytosis. It will help the remnants to enter the liver. And what does the ApoC do? Again, C stands for cutting and cleaving. So it will help in the activation of lipoprotein lipase. So this was same thing which I have told you. This is a liver. This is VLDL gets transformed into LDL. LDL is delivering this cholesterol to the cells, and this HDL is the one which is removing the excess cholesterol, and liver converts HDL into the bile salts. So here you can see VLDL. VLDL has what? More of triglyceride, and it is having less of a cholesterol. So now the lipoprotein lipase enzyme gets activated because given by the HDL, that is the ApoC, helps in activating of this LPL. So because this it will activate, it will cut and cleave it, the VLDL becomes intermediate density lipoprotein, and the intermediate density lipoprotein will become LDL, that is low density lipoprotein. Now LDL will be less in triglyceride and more in the cholesterol. So this is lipoprotein lipase enzyme. This is hepatic lipase. So you can see here that cholesterol ester transport protein. So this will help in the transportation of this cholesterol esters. So what it will do? It will remove triglyceride, and there will be now more of a cholesterol in this LDL. Now, for the LDL to enter the cells, for the LDL to do its function, it needs receptors. So LDL is what? LDL is again a van. It is nothing but a lipoprotein. So this will carry the fat. Which fat? Cholesterol into the tissues, and that when the level of LDL, the bad cholesterol, increases, the tissues are overloaded with this fat. Hence it can cause the risk of atherosclerosis. And for it to enter this, you need the receptor. LDR receptor, which is a protein found on the surface of many cells. So these LDL receptors are there on all the cells, but majority of these are found in the liver. So the binding of this LDL receptor with the LDL, it will cause endocytosis, wherein the cell takes up the LDL receptor complex into the cell. So the LDL receptors are also very, very important. So this LDL receptor, it will help in the binding of the LDL, and this is taken inside by the process of endocytosis. Inside what happens? LDL is broken down and it will release cholesterol, which the cell can use for various function, or it can even store also. The LDL receptor, whatever is there, it is recycled back to the cell surface. So once the work is done, the LDL receptor is brought back to the cell surface. So why this LDL receptor is important? Because this is essential for maintaining the healthy cholesterol level. Suppose if the LDL receptors are not functioning properly, correctly, or if they are absent, the LDL level will rise, leading to buildup of cholesterol in the arteries, and that can increase the risk of heart disease. Sometimes there is mutation of the genes that code for these LDL receptors, and that can cause to genetic disorders which will lead to familial hypercholesterolemia. Okay, so such conditions can happen. Now there was something called as the ApoB100. So now the Apo is apoprotein, and this is B100. So what is this ApoB100? This ApoB100 is a protein which is present on the lipoprotein LDL. So you know that every lipoprotein LDL, HDL, VLDL have a apoprotein in it, which is a brain of this lipoprotein. So similarly, your LDL is going to have one apoprotein that is called as the ApoB100. So now this ApoB100, this is the one which will help in giving the lipids to the peripheral tissues, to the cells. So it will make the cells more and more fat. So this is a protein which is a major component of LDL, and it is seen in VLDL and IDL also, and it becomes important because it helps the LDL to give the cholesterol to the tissues. So elevated levels of this ApoB100 are associated with an increased risk. So whenever there is an increased ApoB100, it suggests that there could be more of an LDL and there could be increased risk of atherosclerosis. So it is sometimes used as a biomarker also. Remember, LDL will give fat to the peripheral tissue, whereas HDL will deliver the fat. HDL will deliver the fat to the liver. So it will clear up. HDL will clear up all the things, all the extra cholesterol, remnants, chylomicron remnants, VLDL remnants, all the remnants are taken by the HDL to the liver. LDL will carry this cholesterol, and this ApoB100 will help it to give it to the peripheral tissue. ApoB100 and LDL receptor, the mutation in both this ApoB100 and LDL receptor can cause familial hypercholesterolemia, which I have already told you. So ApoB100 is thought to be a more accurate indicator of cardiovascular health than the total cholesterol or LDL cholesterol. So sometimes we even do this ApoB100 as a test. So I have also told you that this LDL receptor, majority of them are present in the liver. Number of LDL receptors, particularly on the liver surface, is regulated by the number of cholesterol inside the cell. When the cholesterol levels are inside the cells are very high, the expression of this receptor is reduced. And when the cholesterol level is low, the expression of the receptor is increased, so that it maintains a balance. So this is how I was telling you that this is the liver. Here you can see these are all the LDL receptors here, and here you can see there are receptor for the remnants also, because whatever remnants are there, they will be taken by the HDL into this one. So small intestine, we got the chylomicron remnant entered into the liver. From the liver we found VLDL, that got converted into IDL, that converted into LDL. So then this LDL will be giving the fatty acids or the cholesterol to the tissue. So there is oxidative modification of this LDL, and this will be taken into the macrophages with the help of the scavenger receptors. Whatever remnants are there, they will be cleared by your HDL. So this is how the fat is transported and metabolized in the body. Thank you so much for watching the video. Please like, share and subscribe the channel. Thank you.